Hi everyone, welcome to Foresight's Biotech and Health Extension Group. Really happy to have Wanding Zhou here from Penn Medicine. And today you'll be discussing an updated Infinium BeadChips for delineating DNA methylation dynamics in large cohorts. Thanks a lot for joining us. I'll be in the chat monitoring questions from now on. I'm going to share some more info about you in the chat, and then I'll be starting collecting questions for the Q&A. Thanks a lot for joining. Really appreciate it. Glad you made it. And the stage is yours.
All right. Thanks, Alison. Let me get started. My name is Wanding. It's a great honor to be invited to kind of present some of the work and then some of the updates of the Infinium bead array technologies to this group. As many of you probably know that I'm a computational epigeneticist. I've been working with Illumina, collaborating with them to develop both content and technologies for the Infinium bead arrays for several years. I'm not the original developer of this technology, but I participated in the design of several arrays starting 2015. I think I'm just going to take this opportunity to give everyone, potentially the users of this technology, some updated knowledge of the Infinium bead arrays, particularly the mouse arrays and the EPICV2 arrays, which we recently characterized. So I'll get started. The main thrust of our lab focuses on the DNA cytosine modifications. And then as you probably know, this is a modification to the cytosine base at 5-position carbon. And then the modification is enzymatically deposited by the DNA methyltransferases, the DMTs. There are several versions of them. Some of them deposit the modifications from previously modified cytosines, de novo methyltransferases, DMT3 and 3B. But others, they copy the methyl group from the modified strand, the parent strand, to a newly synthesized strand during DNA replication. And then the reason why I call it the modifications, because I call it also includes other modifications like the 5-hydroxymethylation, which was previously considered the oxidative derivative of the active demethylation process, mediated by the iterative oxidation by the 10-element translocation enzymes TET123s, followed by base decision repairs or TDG actions to fully complete the demethylation cycle. But recently, people realized that the hydroxymethylation is more seen as a separate, kind of stable epigenetic modifications. And then collectively, cytosine modifications plays an important role in many physiological processes, gene transcription regulation, which is most notable. Gene modifications attract a lot of attention recently due to its translational significance. For example, people have successfully leveraged cytosine modifications to build data science models to make chronological or biological age predictions, to classify diseases, to track and monitor progression of these diseases, most notably cancer. And then one of the major motivation behind the use of cytosine modification is that it's pretty chemically stable, and then it can even survive what we call difficult conditions, such as the formalin fixation, paraffin embedding, or even cell-free environments. And the other advantage of cytosine modifications is it's a source of genome-wide signal, not just the genic regions, but also its regulatory informations, the binding, for example, the binding of transcription factors. And then lastly, there is some informatic advantage of studying cytosine modifications in the sense of it has a lytic nature, and hence what allows linear deconvolution of cell quantities for studying tissue composition. Over the years, there has been dozens, if not hundreds, methylation profile technologies developed, some of them based on kind of antibody affinity, and other based on base conversions or direct detections. So this is to show you the focus of today's talk, the Infinium array, where it lands in terms of the number of samples profiled, and then the number of CPGs that it can cover. And actually, I should say this is a figure made by my postdoc advisor over 10 years ago. So more recently, there are other methods have been developed which leverage different, for example, base conversion principles, or can look at other omics as a kind of co-assay, as well as to study single cells. So as I mentioned in the previous slides, the advantage of the Infinium arrays is that it stays in the middle and hit a nice spots of the trade-off between this number of samples provided and then the number of CpGs that can cover genome-wide. And then there are other advantages of these technologies in the sense that it offers a consistent set of CpGs and hence facilitates cross-study comparisons. It's relatively accurate quantitative in the sense that it's shown to be equivalent to a very high sequencing depths if you compare it with sequencing experiments. It is relatively resistant to incomplete bisulfite conversion due to the principle of using hybridization to only target the fully converted, at least in the internal probe sequence, CPH converted genomic DNA. It allows automated sample handling, and then it can reach a relatively high sample throughput. As a result, we have over 100,000 samples, public data deposited to Zhou, which is a gene expression omnibus public repository to host these genomic data sets. And hence, this allows us to trace back and compare previous studies in a more, kind of quite standard compliant manner. And then lastly, it will offer rich information beyond methylation, such as copy number variation and then genetic ancestry. So the way it works is based on this base conversion principle, often mediated by sodium bisulfides, which turns a modified cytosines to a uracil, and hence after PC amplification, a diamine, but modified cytosines are retained to be read out as cytosines. And then this is shared between the array and the bisulfite sequencing. There are two ways that after bisulfite conversion, the arrays actually read out the methylation signal, what we call it Infinium1 and Infinium2. Basically, the way it works is for Infinium1, people have two probes, each for the methylated allele, and each target methylated allele and methylated allele respectively. And then after upon successful bisulfite conversion hybridization, these probes are paired with genomic DNA and then extended by one base, often attached to a fluorophore, depending on the base identity. It could be red and green, and then often it's AT for red and GC for green. And then based on comparing the methylated allele signal, and you're able to derive the methylation state. And then this is Infinium1, and the Infinium2 works by only involving one degenerate probe, and the methylation state is determined by contrasting the two color channels. And this is to leverage the fact that if you are the cytosine being interrogated is fully converted, it will be paired with an A because it's converted to T. And then if it is modified, it will retain it as a C and then be paired with a G. And then in either case will excite the different fluorescent color. After knowing this, we can tell that this is a pretty popular technology and actually most popular for or relevant to study population scale methylation kind of projects. This figure shows the adoption of the 3 consecutive generations of the methylation bead array platform, the HM27, and then the pretty well-known HM450 and then EPICV1. We haven't incorporated the EPICV2 yet because it's recently just released. But as you can see, there is not a leveling of adoption yet, particularly in studies that requires a lot of sample profiling, such as to look at the epigenome-wide association studies, to perform a large number of classifications of disease, as well as to link the methylation variations to genetic variations. So this is, again, the list of multiple generations of the Infinium technology. So as you can see, that's many of the developments over the years. First of all, it started over a decade and a half ago from the introduction of HM27 K-array. But as it develops, it introduced more advanced technologies and also covers more diverse genomic content. And then much of this is reflecting the developing understanding of the methylation biology as well. For example, the HM27 only focus on the CpG island and promoters, but for 50 arrays, they also included the gene body in the recognition that the gene body methylation could reflect the gene transcription status. And then in 2015, when the EPICV1 was released, it has an expanded collection of like targets of the enhancer elements. And then 2019, spearheaded by Steve Horvath and Jason Ernst, they developed an array, the Mammal40 array, using the degenerate-based design to target conserved sequence in mammalian species. And in 2021, we designed the mouse 285K array, incorporated some opposite strand designs. And then in last year, actually, Illumina released end of last year, EPICV2 array, which we participated in the characterization. So in the focus of this talk, I'm going to cover the last two, the mouse 285 array and then EPICV2 array, hoping to give everyone an updated knowledge of some of the kind of novel designs and like content change and other like technical aspects. So this is a typical design flow of how people design arrays. This is extracted from our paper published last year on the mouse MN285 array, but it's largely similar principle for the other array design and developments. So essentially, we started from all the designs, the design candidates of all the CPGs in that species. And then we perform a few quality screenings, such as to ensure that every design have a unique design mapping that is relatively free of the influence by the sequence polymorphisms, and also have a relatively high like extension efficiency. And then after all these screenings, we're often left with half of the CPGs in the genes with feasible designs. And out of these, we perform some CG selections. And then in the mouse arrays, we target certain genomic features. As you can see from the big table on the right, this includes like promoters, enhancers, gene bodies, repeats, sex chromosomes, and mitochondria. And then it also includes 8 like target biology groups with a kind of big focus on, since it's mouse array, on the development, differentiation, like early embryonic developments, germ cell developments. But also we designed it with the kind of goal of building data science tools such as epigenetic clocks, and then to look at cross species, how should I say, to study the conservation of epigenetic mechanisms across human and mouse. And aside from these designs with known biologies, known genomic content features, we also included some random CPGs to retain some capacity of exploring or understanding more unknown biology. And then lastly, we included some kind of utility pros for kind of mouse strain typing and etc. And then usually first thing part of this application is to ensure that the bead array has a high qualitative or technical performance. So from left to right, this is to show you that the array has a high reproducibility by comparing two technical replicates. The array has a high accuracy by comparing it to samples of a titrated methylation fraction. And then lastly, want to make sure that the array is robust under a scenario where the DNA quantity is relatively limited. This is to show, for example, we're looking at 5 nanogram and 500 nanogram input DNA. And then on the y-axis, we're comparing both to 100,000 nanogram. And then on the 5 nanogram, you can see that we have a loss of intermediate methylation, reflecting loss of heterogeneity as you go to very small input. And then again, we also want to validate the array, the new arrays, in terms of its capability of capture biological variations. This is to show an experiment, cell lines in the mice with mutant DNMTs or treated cells that are treated with DNMT inhibitors, in this case 5-aza-deoxycytidine. So as you can see that the wild types, regardless whether it's cells or a mouse, and it's relatively showing a typical bimodal distribution of methylations with a high median methylation level globally. However, the heterozygous cases, they show some slight loss of methylation, but not that noticeable. But most dramatic methylation has been observed in cases of homozygous variants or mutants, as well as compound heterozygous cases. And then interestingly, we are able to recapitulate some of the kind of global methylation difference that's consistent with reported kind of residual methyltransferase activities from the original study that created those cell lines almost 30 years ago. With these kind of tools in hands, I think I mentioned the main advantage of the array over sequencing platform is that it can be applied to robustly assay a lot of samples and hence to dissect intersections of multiple variables in the population, sometimes highly collinear variables. So this is to show you how we use mouse methylation to study cohorts of close to 1,000 or over 1,000 samples of different tissue types, strains, mouse strains, and sex and age. And then we want to ask this question: what factors are the main governing factors affecting methylation patterns? And not very surprisingly, that is tissue type. And then by just simply projecting the methylomes to a 2D plane in an unsupervised manner, we can see that the tissue types drives the clustering pattern. And then this is followed by, I think, the age and the experimental group strings. But if you look at these specific methylation changes, for example, a strain-specific, sex-specific, tissue-specific, and age, you see an extensive amount of statistical interactions. And then, interestingly, mouse strain was also shown as a pretty major determinant. Just to zoom on the strain a little bit further, we can even link the strain-specific methylation difference to the strain phenotypes. For example, in this case, BTBR mice, which is autism model, we're able to identify some neuron-specific or the brain tissue-specific loss of microimprinting at this site of HERX3 and the NAP1L5 loci. And then again, we were able to find some loss of methylation in the LPJ mice, which has a neurocrest development issue. And then turned out that most of the hypomethylation goes to the binding sites of factors that are mastering the neurocrest differentiations. And then potentially that kind of mouse has a kind of seizure phenotype. So we start speculating that it could potentially have an implication there. And like I said, you can possibly dissect highly collinear cases. In this case, we are looking at the age and then the cancer, because cancer is considered age-related disease, and both processes involve a lot of cell proliferation. And hence, it could be difficult to fully disentangle the two processes. And then the methylation changes coupled to the two processes. So in this study, we actually it's the same data sets. We look at dozens of samples with various age and then oncogenic tumor status, malignant status. And then we perform multiple regressions, and we found CPGs that are coupled to age and then coupled to tumor, but not to the other. So in this case, you can see that the age-associated hypermethylations are primarily located at the CBD islands and polyclonal targets, while the tumor-associated methylations, hypermethylations, goes to enhancers. And then zooming into the actual binding sites of these age-associated hypermethylations, you can see that it goes primarily to polyclonal binding, such as SUSI 12, EZH2s, and then the hypomethylation, on the other hand, goes to the bindings of the cohesing complex members, potentially like indicating a alterations in 3D conformation changing as the mouse age. And then on the other hand, the cancer-associated methylation gains goes to binding sites of transcription factors that are master regulating the corresponding tissue. In this case, intestinal tumor, so it's mostly intestinal cells, the colon or intestinal mucosal cells. And then these modifications also regulates the genes that are important to the function of the intestinal epithelium cells. And then if we look at hypomethylation, on the other hand, it goes to binding sites of AP1 transcription factors, which is extensively implicated in differentiation, proliferation, and apoptosis, and then showing a cancer link. And then with these, you can also build a mouse epigenetic clock. And then again, we're showing that epigenetic clock the selected features are enriching these polyclonal targets. So this is what I want to show you about the mouse arrays. Next few minutes, I wanted to talk about the EPICV2 arrays. And then it is an expansion on top of the EPIC array, V1 array, and then the HM450 array. 83% of the EPICV2 or EPICV1 arrays is inherited to EPICV2 arrays. And so these ensures very good backward compatibility. And a major issue of EPICV1 is that, and then HM450 or any other previous Infinium array for that matter, is the influence of the human polymorphism, probe hybridization, and extension. This could include kind of SNPs that go to the internal sequence and affect probe hybridization, or hits the extension base and hence alter the color channel measurements, and then or even just duplication deletion of the whole region, and hence altering the hybridization, and hence leading to a potentially misleading pattern of epigenetic silencing, as shown in our previous work. So the EPICV2 was designed to address this or alleviate this. So you can see that the cross-reactive probes were excluded in the EPICV2 design, and also like for most of the kind of ancestry-specific SNPs, their influence on the EPICV2 is mitigated compared to EPICV1. And also EPICV2 has fewer kind of poorly mapped probes, all based on this empirical learning of the performance of EPICV1. And then in terms of content, EPICV2 includes most of the additional CpGs to focus on enhancers, tumor classifiers, epigenetic clocks, and the EWAS hits. So this is to show the relative enrichment of these, and then also the mapping with a recent kind of methylation reference coupled to cell type. And then also captures the existing kind of clocks pretty well. It doesn't retain all the clock probes, but most of them, and much more enriched than the array average. And then this is to show you the consistency between EPICV1 and EPICV2 on 4 different cell lines. And in fact, that you can even see that the EPICV2 only probes, they retain the cell discriminative power very well, and also has a certain level of toleration of DNA input variations. And then EPICV2 also uncovers the global methylations in the Dnmt cell lines. This is to show you a validation of this biological variation in the HCD116 cell line and its derivatives with knockouts or hypermorph either Dnmt1, Dnmt3A, 3B, or SETD2 genes. And then you can see that the DKO1 cells with a hypermorph Dnmt1 and the loss of Dnmt3B shows the greatest loss of methylation, while the Dnmt1 KO, 3AB DKO, they do not have significant loss, but they do have some noticeable loss compared to the wild type. And then it also shows, for example, the DKO retains the methylations, goes to imprinting regions and transposable elements, and they suggest a critical role of these elements in ensuring kind of minimum cell viability. And then, like I alluded briefly, the EPICV2 has a great low input performance. In this case, you can see that on the right, we have 250 nanogram all the way back to, we tested to 1 nanogram, which has greater than 50% probe detection rate. And then if you cluster the methylomes based on the input, which is shown on the labels, you can see that the samples primarily clustered by cell identity instead of by DNA input. And then this is the case even for EPICV2 only probes. And most of the probe detection loss goes to the quiescent and heterochromatic regions. And then the CG-rich, the GC-rich bivalent chromatins or promoters, they tend to retain more detection. And then lastly, this EPICV2, the Illumina has developed some new category of probes called NV probes. These are included to target 64 unique genes. These are genes known to be recurrently mutated in cancer. In this case, you can see that, for example, we tested on the 4 cell lines, and one of which has a signature KRAS G13D mutations, and it was correctly captured on the array. And also, just like the EPICV1 and the HM450 arrays, the EPICV2 can be pretty readily used to study copy number variations. And then this experiment is similar to the titration experiment that I showed in the mouse array, but this is to show you that the most majority of the CG probes are highly correlated with the titration and methylation levels, but not the CH and CTs and other probe categories. And actually, we are providing this list of suboptimally correlated CGs so that user could reference for potential artifact detection. And the most majority of these poorly correlated CG probes have a relatively small beta value effect size. It is also enriched in the mass that we are providing as part of this characterization effort, suggesting a residual polymorphism influence, causing this loss of correlation. And then lastly, I just want to show this because this is a frequently asked question to people about this new probe ID that is introduced to EPICV2 and the mouse array. Because previously the CG number is 122 mer. It's not a unique description of the probe sequence. So we added those suffixes, and then those suffixes could indicate the fully described design, including the top and bottom and which strand is bisulfite converted. And then the replicate probes has a high interprobe correlation compared to the non-replicate probes. And in summary, the EPICV2 and then 285 are robust, reproducible platforms. And then we have applied it to study methylation variations in large sample cohorts. And then we show that it can be used to dissect even collinear variables in these cohorts, such as age and tumor. And then the updated EPICV2 can be applied to more diverse human populations, and it could even serve as a multi-omic tool for cancer genomics. So this is just to thank all the contributors to the characterization work and our collaborator from True Diagnostics, and Ryan, our collaborator from Illumina, who supported the design, and also my postdoc advisor, collaborator from Foxel Technologies, and also our collaborator Rahul and Marisa from Penn, who provided some sample access supports. Yeah, happy to take questions, and thanks for listening.
Awesome. Thank you so much. Really appreciate it. This is a time for anyone. If you have a question, feel free to raise hand, or feel free to ask your question here in the chat in case you cannot unmute. But I'm super curious just to see where, if you're thinking like 5 years ahead or so, where could you be ending up with the kinds of tools that you're currently developing? If everything goes well, where do you think we could be in about 5 years or so?
Sorry, can you repeat that question again? I didn't quite catch her.
Oh, sorry. Yeah. If things go as planned, where do you expect us to be able to be in 5 years or so if tool development and so forth goes just according to plan?
So you're talking about the tool development the assay development or the informatics development?
Both.
Both. Okay.
If your entire project just goes according to plan, then where would we be in around 5 minutes or so? In around 5 years. 5 minutes would be a little bit too early.
Yeah. So I think there's many things going on in the field. So there's a dropping the sequencing costs, which are closing the gap between the arrays and the bisulfite sequencing, which obviously make the cost-competitive arguments weaker. But I think there are other aspects of the array which is still important to keep in mind, such as its quantitative performance, its resistance to the incomplete bisulfite conversion, and also the presence of the legacy data, which allows this data integration. So I think it's reasonable to expect the field and investing more effort in integrating the two platforms as we go forward. For example, the array developments will take advantage of the knowledge acquired from, for example, the single-cell methylome sequencing, while the sequencing projects could take advantage of the legacy data that previously generated on the arrays. But I think a major challenge here is how do we discriminate what is platform-specific artifacts and what is real biology? Yeah, I think the field is moving forward to address these questions. In terms of the method development, I think there's a lot of exciting developments in data science that kind of are leveraging the vast kind of abundance of the array data out there. And then I think, but there are some, I think it's the same challenge that I was mentioning, which is how do we develop these platform independence, robust methods, right? So when it comes to, for example, age predictors or the cancer classifiers, so that we don't have to regenerate all the data using updated technology. Yeah, that's just some thoughts. I don't have a very good answer for.
No, that's only this is
Our
like speculative question. It's just really fun to hear, I think, where this could be going. It's really. Next one up, we have Aram, in case you can unmute. Otherwise, I'll ask you a question.
Yeah, again, my name is A. Ramo. Ramo is the last name. I didn't figure out how I can put the O there yet. So my question is, you concentrated on the methylation chemistry, and I think this is wonderful and very interesting. We're still trying to understand the consequences of these modifications on the DNA. But there is also a whole array of other chemical modifications on DNA. And I'm not going to touch the RNA modification right now. I think it's beyond the subject matter. But there are other modifications, even if those modifications are not stable and they have a shorter time period and they prone with instability, but still they may actually contribute to the whole theme of different expressions. Can you tell us more about what is the expectation about how much the story about DNA methylations occupies in general vis-a-vis the chemical changes and the DNA? Can we be comfortable that studying DNA methylation will tell us most of the story, if not all?
Yeah, I think that's a great question. My impression is that this depends on the biological context. For certain biological contexts, for example, like even just for, let's say that there's a species context, there's a tissue context, right? The cell type context and such. If we just think about like a human, probably there's not so much, for example, or it's still disputed whether we have like M6A or other like modifications that are more abundantly seen in bacteria. Because bacteria is a rich resource of different DNA modifications to all different bases, right? All 4 bases. But for kind of particularly higher order eukaryotic species, I think more focus is still on the cytosine methylations and its derivatives. But there are growing appreciation of, for example, 5-hydroxy methylation, and then how we can use similar technologies but different conversion kind of principles to query them, like even to base resolution, and then to study them on the same molecule with the 5 MCs. And by being more transient, probably you are talking about 5 carboxyls and formylcyls. Yeah, these are relatively understudied, and I think my knowledge is limited to conclude that how much role they play. But I think I would imagine they do vary from cell type to cell type, and from like one genomic territory to another genomic territory, like a base sequence composition context.
Yeah. Thank you very much.
Awesome. Next one up, we have Fiona.
Yes, thank you very much. This is remarkable stuff, very interesting. I am just curious about just the measurement technology. How much difference do you expect from lab to lab? How do you control the error sources? And also, if you can speak to the whole measurement setup for a variety of measurements and the costs and how much this technology can proliferate.
Yeah, I think that's actually one of the advantage of the Infinium beta arrays, and namely that it can, it's relatively considered more based on experience, not on any kind of rigorous quantification, or based on experience. I think arrays are more robust when used in like different labs. Actually, this allows like one, it always gives you the same scope, like in the sense that the same set of CpGs are being measured, and then 2, it has a relatively high equivalent read depths on the arrays, And so it's more quantitative. And I think more reproducible data has been seen with the arrays across labs and compared to sequencing counterparts. But I think the sequencing is catching up, particularly with the capture MesoSeq platforms that allows you to kind of look at a small limited set of CpGs, but to a very high depth, and it's achieving the same thing as arrays.
Is there any calibration involved?
Yeah, actually, this is a great suggestion because this is something that we are thinking about as well. So right now, the calibration has not been systematically done on the arrays. Well, from the community side, I'm sure that the Illumina has internally, they have done lots of calibrations. Part of the challenge is that, actually also mentioned that the arrays, it has some intrinsic shortcomings. For example, you will never be able to get a good quantification of the extremes, like 0%, 100%, due to the presence of signal backgrounds. You can dichotomize it, or discretize it so that you infer maybe any value that is smaller than 5% to be 0%, or 95% to be 100%. But that is the raw readouts doesn't give you that information. A potential solution of this is to derive a standard curve, like for example, using kind of titration data. And then I think this is something that could be done, and maybe Illumina has already done it internally. But yeah, there are some, I think, and also there's some challenging that come with those calibrations, because the application scenario with DNA methylation arrays is so diverse. And if you do such calibration on cell lines, it might not work on primary tissues, primary cells. Yeah, if it works on one tissue like blood, it might not work on like other buccal cells or other brain tissues. So that might be the reason why the calibration, like a full calibration, might be challenging. But yeah, I think that's an important point. I agree.
Yes, thank you. And can you comment on the cost? How much it is a setup?
How much is the cost to run this arrays?
Yeah, the cost to set it up and run, just to acquire.
Yeah, so I think the DNA preparation, so if you don't count the extraction parts, like maybe bisulfite conversion, plus I think it depends on which core you use, but I think the price ranges for EPICV2 ranges from like $300 to $500 per sample.
What about the actual lab setup, the equipment?
This one needs the night scan. If you don't have, usually the iScan works for both the methylation bead arrays and the the Illumina SNP arrays. So any genotyping course will have this. And if you don't have the iScan, I was told it could also work with the Nexi 500, but yeah, no personal experience.
Okay. Thank you very much.
Yeah.
Cool. Next on, we have Jyoti. If you can, otherwise I'll ask you a question.
I have unmuted. Thank you, Alison. Thank you so much. This was a very interesting talk. So my question is more from an understanding perspective. So in one of your earlier slides, you talked about the target biology group, which talked about various things you're looking at. Question one is, are these different arrays that you're creating? Are all these embedded within one array? The related question there is, I would love to understand a little more on this human mouse synteny group CpGs that you've included, the significance of that especially.
Sure. Yeah. The short answer is it's one array. So it's a different selection groups for probes for that one array. And also a lot of these selection groups, they overlap. So it's not that they are different CpGs, completely like mutually exclusive CpG sets, but they were selected. So you can see that the number doesn't add up to the total number. So it's overlap removed. So the second question, the human mouse synteny. Yes, although mouse is quite remotely related from human, but it's one of the most extensively used model organisms to study human disease. That's the main motivation behind looking at the human mouse conservations or differences in methylomes. So we designed those probes so that you can apply a human array like a EPICV2 and a mouse array And then some of the CPGs can be directly compared in the sense that they goes to the same genomic regions, the same gene, the same, for example, position with respect to that gene promoters or UTRs, first exons. And then they are considered the equivalence like between the two species. And hence observations made on a mouse experiment could be potentially extended to humans. So that's the main kind of motivation of including those. And there are about, I think it's close to 20,000 CPGs that has this properties of direct mapping.
So I think I'm going to reframe my first question. I understand this can all be in one array, but from an application perspective, see the early development one, could it be a separate chip altogether so that it could have a specific application?
Yeah, I think it can certainly. But I think there's also a kind of cost consideration. That's more of a kind of Illumina's business decision, because to build an array, there's a upfront cost, and then I think it's very hard to build an array for a small group of investigators who's interested in one biology. That's why actually this is also the case for EPICarray. The beauty of this is that you can generate data for one biology, and then if you someday in the future you're interested in a different biology, you can reuse that data. So this is considered a genome-wide assay instead of, for example, a kind of early development embryo assay. But I think there are some custom arrays. This is I probably should ask Illumina more directly. I think Varun also knows that better. Like you can custom make some arrays for just, for example, imprinting biology.
Mm-hmm. Okay, thank you so much.
Okay, awesome. We have 3 minutes left. You can stop me if you have another question. Otherwise, one thing I'd be super interested in hearing is, do you have anything, if there's let's say early stage researcher that's listening to this, gets really inspired, is there a particular area that you would encourage them to solve? Is there a particular bottleneck that you think you guys will be facing that you would love some help with?
I'm an early stage researcher. No, just kidding. Yeah, I am an early stage researcher. If I would have any suggestion for myself, I will probably reiterate that kind of gap where I see, for example, there's a platform specific measurements variations. I think that needs to be discriminated from the real biology. And then particularly now that the technology is being updated so fast, but the data generation actually takes time. And then the biology and the kind of discovery takes time. So how do we reconcile the two, right? So how do we apply a technology and then expect the data to have a long lifespan? So I think this is something that me as a technology developer can think pretty often.
Awesome. Thanks. Final question. What can people in this group help to advance your work in particular? Is there anything that you are hoping to do in the next few months or so that people in this group may be able to help with? And maybe looking for collaboration, or you're looking to fundraise another round, or whatever it may be. Now is a great time to plug it.
Yeah. Just one thought that runs through my mind is that we are developing a new array, a human array, that has a more concentrated selection of the human trait associations. This is going to be a smaller arrays, and we're definitely looking for thoughts or collaborations or ideas or feedbacks, and we are hoping to release with the Illumina this array close to the end of the year. And then a lot of these focus is going to be on the cell type deconvolutions and using the arrays to track cell identities. Yeah, that's if we have an opportunity, we would love to kind of receive feedbacks or maybe collaborate on the future development of the Infinium bead arrays, particularly with labs that has maybe a kind of independent, for example, sequencing, technology development backgrounds.
Awesome. That is pretty actionable. Thanks, everyone. Thank you so much for joining. We really appreciate that you joined us. This was wonderful, and we'll have the recording up soon. Thank you, everyone, for joining as well. And I see you very soon for the next one. Bye, everyone.